At this point in Chapter 2, we move to the topic of biological molecules. So we've talked about how bonds are formed, we've talked about covalent bonds, ionic bonds, and hydrogen bonds, and now we want to utilize that information to understand how the molecules that compose you are put together. So much of this story revolves around carbon. So you know at this point that carbon has an atomic number of 6, which means that it has a total of 6 protons and 6 electrons. 2 electrons are found in the first shell, as we see here in our slide, and 4 electrons are found on that outer shell. Furthermore, at this point, we remember that in order to be content, carbon wants to have a full outer shell of electrons, and so that means that it will go and share electrons, forming covalent bonds with 4 partners. And so when it forms those covalent bonds, as you remember from our previous discussion, it ends up with a total of 8 electrons in its outer shell because it counts the electrons that it's sharing as its own, although it's actually sharing them with other atoms. And so we end up with a structure that looks like this, what we have on the bottom of our slide here. The central atom is a carbon atom, and it's forming bonds with 1, 2, 3, 4 partners. The rod shape that we see here represents the covalent bond, the pair of electrons that's shared between carbon and another atom. And so biological molecules are all carbon-based molecules, and the biological molecules tend to be very large, extremely big molecules, because they are long chains of carbons, shown here at the bottom of our slide, with other atoms attached to the sides. So for instance, this carbon atom in the middle of this chain that I'm pointing to with the cursor has 4 partners. This carbon is a partner, this carbon is a partner, this hydrogen is a partner, and this hydrogen is a partner. And so you can see that it makes sense that carbon would form these long chains with little sidebars coming off periodically. And the molecules of your body are mostly very large molecules, and they're mostly carbon-based molecules. And that's our goal in this particular section of Chapter 2, is to talk about biological molecules, which are carbon-based molecules. So we have 4 different types of what we call organic compounds. Now, when I use the term organic molecule or compound and biological molecule, then I mean the same thing. Organic molecules are biological molecules. And there are 4 types. There are carbohydrates, lipids, proteins, and nucleic acids. Let's move on to carbohydrates. So carbohydrates are known as sugars and are composed of varying short chains of carbon atoms with hydrogen and oxygen atoms attached to that. So the very simplest carbohydrates are known as monosaccharides. Monosaccharides, we said that most carbohydrates are composed of short chains of carbon atoms with other groups attached. And so monosaccharides are composed of very short chains, 5 to 6 chains of carbon atoms. Disaccharides are 2 or more, actually disaccharides are just 2 monosaccharides that are put together, and then polysaccharides are 3 or more monosaccharides that are placed together in a chain. So here's a monosaccharide. So this is glucose that you're certainly familiar with. You're alive right now because there is sufficient concentrations of glucose present in your blood. And we see 1, 2, 3, 4, 5, 6 carbons in the backbone of glucose. And now this doesn't look like what we saw before, does it? It doesn't look like a long straight chain. That's because glucose curls up on itself. And so it curls up in aqueous solutions and forms this ring structure that we are familiar with. So that's one example of a monosaccharide, but there are many other monosaccharides. And what we'll see is that monosaccharides are the building blocks of disaccharides and polysaccharides. So here's a disaccharide. This is sucrose, common table sugar. So when you go to the store and buy a 5-pound bag of sugar, this is what you're purchasing, sucrose. And as you can clearly see here, sucrose is composed of, sucrose is composed of 2 monosaccharides. We have glucose on the left-hand side, and then we have fructose, another monosaccharide, on the right-hand side, joined together by a covalent bond. So this is what our larger carbohydrates are going to look like. They are going to be chains of monosaccharides. A monosaccharide is composed of 5 or 6 carbons wrapped around itself in a ring, and then disaccharides and polysaccharides are long chains of monosaccharides. So we move on to polysaccharides at this point. Did I say that correctly? So I meant to say disaccharides and polysaccharides are long chains of monosaccharides. Here we have polysaccharides. Polysaccharides are composed of many different monosaccharides, not just 2, but long chains of monosaccharides. So examples include cellulose, starch, and glycogen. This is cellulose, and so cellulose is a very common polysaccharide. It makes up the hard components of plants, and so the 2x4s that you buy at Lowe's are composed of cellulose. Much of the leaves of plants are composed of cellulose. When you eat lettuce, you are consuming cellulose. Starch is a different polysaccharide, but a polysaccharide that plants use to store energy. Glycogen is a polysaccharide that you use to store energy. And so you can see that at least in the case of cellulose, and it's the same for starch and glycogen, that these polysaccharides are long chains of monosaccharides that are all connected together. In fact, there may be hundreds or thousands of monosaccharides that are bound together in these long chains in polysaccharides. We move on to lipids at this point. Lipids are interesting biological molecules, and lipids are somewhat smaller than the other 3 groups of biological molecules that we'll talk about. Lipids also vary quite a bit in appearance. The unifying feature of lipids is that they are all insoluble in water. No lipids dissolve in water. So we have 3 types of lipids that we'll talk about. We have fats, sterols, and phospholipids. So the first we have on this screen here are fats. And so fats are also known as triglycerides because they have 2 parts. They have a glycerol head that I'm circling here, and then they have 1, 2, 3 fatty acid tails. And so those fatty acid tails are composed of long chains, which is what's being depicted here, of carbons with little hydrogens projecting from those. And so don't be confused, if perhaps you are, by this portion of the diagram. These dashed lines mean that what we're seeing here that I'm circling is the same thing that's making up the chain down here in the case of a phospholipid and up here in the case of a fat molecule. So this is a fat is a long-term energy storage molecule utilized in humans and all animals to store energy. Cholesterol is another type of lipid, and so cholesterol looks a lot different than our fat molecule. It consists of 4 rings and has some other groups attached to those rings. Each ring is composed of glucose molecules, of carbon molecules. Each corner represents a carbon molecule, and so this is a long, not super long, but 1, 2, 3, 4, 5, 6 carbon chain that's wrapped around on itself. And so we have 4 of these chains. This one just has 1, 2, 3, 4, 5 carbons, and these rings have been placed together to form this molecule that we know as a sterol. Cholesterol is an example, and a very common example, of a sterol, but there are other sterols that are found in the natural world. Cholesterol, as you know, is found in your blood, and it's also an important part of the plasma membranes of cells and helps form the correct consistency of the plasma membranes. Phospholipids are yet another type of lipid, and so phospholipids are a little different. They consist of a head that contains glycerol and a phosphate group, and then they contain 2 fatty acid tails. And so there's glycerol in the front and then 2 fatty acid tails, glycerol with the phosphate group attached and 2 fatty acid tails. So in just a moment, when we talk about the plasma membrane in this chapter and in chapter 3, we'll be talking about phospholipids. Phospholipids are the most common molecule that's found in the plasma membrane. So those are our lipids. We move on to proteins, and so proteins are a fascinating type of biological molecule. And proteins are long chains of amino acids. And so one thing that we see in the case of biological molecules is that biological molecules are typically polymers composed of monomers. This is not the case for lipids, but it is the case for carbohydrates. Think back to carbohydrates and remember that in the case of carbohydrates, polysaccharides were long chains of monosaccharides. The monosaccharides are the monomers and the polysaccharide is the polymer. So in the case of proteins, the monomer is the amino acid and the polymer is the protein itself. This is a single amino acid that I'm pointing to in part A of our figure. Each amino acid contains a central carbon atom, an amino group, a carboxyl group, a hydrogen, and then a side group that makes up the rest of the molecule. And so it's the side group that's different in each of the amino acids that's present in living systems. The side group gives the amino acid its unique characteristic. Either it's acidic or it's basic or it's polar or it's nonpolar. And so all these other parts, the amino group, the carboxyl group, the hydrogen atom, the central carbon atom, those are the same in all of the amino acids, but the side group is different. So there are 20 amino acids that are present in living systems. There are 20 amino acids that make up the proteins found in your body. A protein then is composed of the linking of these amino acids. Valine is an example of amino acid. And in this case, valine is linked to alanine, which is linked to phenylalanine. And eventually we end up with this long chain. There's typically hundreds or maybe 1,000 or even a couple thousand possibly amino acids in length. The protein curls around on itself and amino acids interact with each other to maintain the three-dimensional shape. So for instance, this amino acid is interacting with this amino acid. And fascinatingly, the shape of this protein is just astoundingly perfect. It looks like a random twisting and bending that we see here on this screen of this amino acid chain, but in reality that shape has to be exactly right in order for the protein to function appropriately. And so that's going to be an important theme in our class. Proteins do all sorts of things in the body. They digest food, they move ions, they transport oxygen, they serve it as molecules that move other molecules from one, such as glucose, from one part of the cell to another. And they have to be formed just right in order for them to function properly. And so the formation of a protein is all about the shape of this amino acid chain. Here's an example of a protein. This is hemoglobin, which is the protein that's present in your red blood cells. There are over 1 million hemoglobin proteins inside each red blood cell of your body. And one red blood cell has over 1 million hemoglobin proteins. And so this is an incredibly common protein in your body, and it's incredibly complex. There's about 450 amino acids, 450 little building blocks that make up this protein. And so that shape that you see on the screen has to be exactly right in order for hemoglobin to bind to oxygen. Each hemoglobin molecule can bind to 4 oxygen molecules and transport oxygen throughout your body. You may say, well, I don't see the amino acids in this diagram, but keep in mind this is a more realistic diagram, and so it would be impossible or very difficult for the authors to draw 450 little amino acids. And so they have chosen to draw a little, you see the spirals found in the different parts almost everywhere of this protein. These are referred to as alpha helices. An alpha helix is a type of protein structure in which the amino acid chain curls around and around and around on itself. And so what you're looking at is a long chain of amino acids. They just haven't drawn the individual amino acids. And so note how perfect that structure is. This is so tiny, and that structure has to be exactly right in order for this molecule to actually function. It's really amazing to think about it. It's an idea that we'll come back to later in our class as we talk about various diseases. Sickle cell anemia, for instance, is the result of a problem in the shape of this hemoglobin molecule. We end our discussion with perhaps the most interesting biological molecule, which are nucleic acids. Nucleic acids include DNA and RNA, and fascinatingly, this is the molecule that makes up who you are. This is the molecule that contains the code that allows you to be produced. Remember, you could, this has hopefully not been done, but it's possible we can clone animals, so we can clone humans, so you could make another you by just having the nucleic acid in one cell of your body, and another you could be created from that. The blueprints to produce you are found in the nucleic acids of your body. Now, nucleic acids are composed of nucleotides, and nucleotides are composed of 3 components. And so on the screen we see from our text the components of a nucleotide. So every nucleotide is composed of a phosphate that we see on the screen here, a sugar that we see right here, and then a nitrogen containing base. And so DNA is the nucleic acid that would be made up of the nucleotides we see listed on this screen. So there are 4 possibilities in DNA for the nitrogen containing base: adenine, guanine, thymine, or cytosine. So let's go on to the next slide and take a look at how this fits together. Now, I will back up and cover the slide that we missed, that we skipped there. So what we see is that the parts fit together in this manner. Focus on the left-hand side of the screen for just a moment. So if we start here at the upper left-hand corner, we see a sugar, a phosphate, and a nitrogen containing base. What I'm circling right here is a nucleotide. This is another nucleotide. This is another nucleotide. And so it's easy for you to see that the nucleic acid, DNA in this case, is composed of 2 chains of nucleotides. And the chains are made of a sugar bound to a phosphate, sugar there, phosphate there, within a nucleotide. And then between nucleotides, this sugar on this nucleotide is bound to this phosphate on the other nucleotide. And so these are covalent bonds, very strong bonds that hold this chain together. There's also a covalent bond that's holding the base onto each of the nucleotides there. But you know that DNA is composed of this structure referred to as a double helix. It is a double helix because there are 2 chains, as you can see, one on the left-hand side, one on the right-hand side, and those 2 chains are held together by these dashed lines. The dashed lines represent hydrogen bonds that go from one base to another, holding those bases together. Hydrogen bonds, as you remember, are very weak bonds, which is important for the overall function of this molecule. Now let's take it one step further and look at the right-hand side of this page. And so on the right-hand side of the page, we see all of the parts we just talked about. And so here's a sugar, a phosphate, and a nitrogen containing base. And so you can follow those all the way down. And so there are 2 chains here. Here's one that I'm running the cursor along there, and here's another chain that I'm running the cursor along there. And those chains are held together in the center of the molecule by the hydrogen bonds that connect the 2, what we refer to as complementary bases. Adenine always binds to thymine, and cytosine always binds to guanine. A with T and C with G. And that's always how those bases are put together. So that's the overall structure of DNA. Everything that is part of your phenotype is coded for by the DNA molecule. And this is the blueprint that forms who you are. Everything that's needed to make you is found in this amazing blueprint in your cells. Let's back up and talk about this for just a moment. Before we mention the cloning at the bottom, let's mention this. And so DNA, what we've been talking about, is double-stranded, and it's the same in all cells of the body. Complementary base pairing is what we just mentioned. So A goes with T, C goes with G. That is the idea of complementary base pairing. A is complementary to T, and they form a bond. C is complementary to G, and they form a bond. So DNA is the same in every cell of the body. That's an amazing aspect of biology. So the DNA in the skin cell, and the liver cell, and the hair cell, and the nerve cell, all that's exactly the same. And so that means, fascinatingly, in order to clone these cattle, for instance, these are all cloned cattle, all they needed to do is take a cell from the cow they wanted to clone, and it could be any cell. It could have been a cheek cell, or an udder cell, or a skin cell, anything from that cow, any cell, and had enough DNA, the complete set of DNA, so that they could produce these little clones that we see on the screen. These are cloned pigs that we see here. Cloning involves taking, and we'll talk about this in more detail in our biotechnology chapter, but cloning involves removing a cell from one animal and then taking the DNA out of that cell, putting that DNA into another egg of a second donor organism from which the nucleus has been removed, and then that egg begins to develop. Of course, no sperm is needed because the DNA is present in what we call a diploid form. We'll table that term for now. We will come back to that and learn it. But that in a nutshell is cloning, something we'll talk about in much more detail with some nice pictures later in our class. So as we leave this topic then and finish our discussion of biological molecules, we want to mention that there's one other type of nucleic acid called RNA, ribonucleic acid. And so the differences between DNA and RNA are as follows. RNA is typically single-stranded. It's usually single-stranded, and it's copied from DNA as it's needed based on the cell type that's found in the body. And so, for instance, you can ask the question, why does a liver cell look different than a nerve cell? And the answer is that different parts of the DNA are read and copied into RNA and then made into a protein. Now, this is an idea that we're going to get to, and a very exciting idea in biology in a couple chapters. But for now, I think it's sufficient to say that we read the DNA, turn it into RNA, and then that message that's formed in the RNA goes somewhere else and does something else inside the cell. It's also interesting to note that RNA, R stands for ribo, and so the sugar in RNA is ribose. The sugar in DNA, as you remember, was deoxyribose, but the sugar in RNA is ribose. So this concludes our discussion of biological molecules.